Mhm. All right. Okay. Hi, everyone, and thank you for joining today. I'm Tanina Arab. I'm a postdoctoral fellow at the Johns Hopkins University, and I'm going to moderate this session with my colleague Yonis Burr, who is a postdoctoral researcher at the Institut de Recherche en Infectiologie in Montpellier, France. We are really pleased and honored to welcome Professor Hendrix. I don't think Professor Hendrix needs an introduction, but just for the sake of the recording, we're going to make one. So An Hedrix is a full professor at Ghent University and a group leader at Cancer Research Institute in Belgium. She obtained a Master of Science in Bioscience Engineering and then a PhD in Health Science. She's organizer of the EMBO EMBL practical course in extracellular vesicles from biology to biomedical applications, and also president of the Society for Extracellular Vesicles in Belgium. During the BESEV 2021 meeting, she was awarded the Early Mid-Career Investigator Award, a very well-deserved one, for her seminal work on EVs, providing supportive ecosystem for their clinical applications, including development of the EV track knowledge database that we're going to hear more about today, as well as SOPs for analysis of EVs in liquid biopsies and reference material. And this is really a very short biography, I have to say. So there is much more to say about all your effort in the EV field. So thank you again, Professor Hendrix, for joining us. We are really honored. You can go ahead and share your slides if you want. Thank you so much.
Thank you for this very kind introduction. And I also first want to thank the shares from the SNF network for this opportunity to inform you a little bit more today on the EV track knowledge base. So let me first share my screen. And this, I think, should be fine now.
Yeah, it looks great.
Okay. So today I want to discuss and go a little bit more into depth with you on the EV track knowledge base, which is a freely accessible online tool that we developed to really stimulate transparent reporting, but also to centralize knowledge on extracellular vesicles. Now, first I want to delve a little bit more into why, or the reasons why we developed the EV track knowledge base. So as most of you probably are aware, the field of extracellular vesicles encounters a lot of heterogeneity. And this heterogeneity is introduced at multiple levels. So extracellular vesicles are secreted by different types of cells, including both eukaryotic and prokaryotic cells. The composition of these vesicles and the type of vesicles that are released depends upon the status of these cells, on the biogenesis pathways through which these vesicles are produced. And this, of course, results in vesicles that differ in their structure, so they vary in size, density, surface charge, and refractive index, but they also widely differ in their composition in terms of proteins, for example, and nucleic acids. On top of that, once these extracellular vesicles are released into their environment, they can also develop a biomolecular corona, meaning proteins or other molecules that are abundantly present in these sources can adhere on the outer side of these extracellular vesicles. Of course, most of the times, when we want to analyze the content, the composition, or the function of these extracellular vesicles, we have to capture them out of these different types of sources. And to do that, we make use of methodology. So first, we try to isolate or prepare the vesicles from these different sources. And then we use also different types of methods to characterize these extracellular vesicles. And also, depending on the methods that you use, you add an additional layer of heterogeneity. So for today, I want to mainly focus on heterogeneity that occurs at the methodological level. So once we decide to analyze extracellular vesicles in a biofluid, most of the time we select preparation methods. So methods that we use to isolate the vesicles from their source. These methods typically exploit different biophysical or biochemical characteristics of extracellular vesicles. And depending on the method that we use, these methods will result in a concentration of vesicles from the sample of interest or a further enrichment of vesicles from the sample of interest. So typical characteristics that are exploited are, for example, the size. So methods that enrich, for example, extracellular vesicles based on size, or differential ultracentrifugation, size exclusion chromatography, or asymmetric flow field flow fractionation. Another biophysical characteristic that is commonly exploited is their density. Different types of density gradients can be performed in a top-down setup or in a bottom-up setup, or for example, also density cushions. Besides that, also some surface characteristics can be exploited. So for example, the presence of particular antigens that are then captured making use of antibodies, or also surface charge can be exploited to separate extracellular vesicles from their source. So a wide scale of methods that can be used to prepare vesicles from a sample of interest. But one important question and one important consideration to make is, first of all, does the choice of method that you use to prepare vesicles from the sample impact the downstream analysis? For example, in this particular case where we were interested in the identification of biomarkers. So here we started from a very simple biofluid, conditioned medium, which was produced by breast cancer cells. We collected the conditioned medium and concentrated the medium so that we were able to prepare vesicles making use of three different methods: a density-based method, a size-based method, and then also a commercially available method which exploits polyethylene glycol as a volume excluding polymer to precipitate biological materials from a sample. From these three preparations, we then analyzed the RNA composition. We actually observed that the RNA composition was really different and dependent upon the method that we used to isolate the vesicles from these samples. Interestingly, the RNA composition that we obtained with the density-based method significantly differs from the RNA composition that we obtained making use of the size-based method or the volume excluding polymer. By doing a further downstream analysis in that particular study, we actually observed that only by making use of the density-based method, we were able to differentially enrich extracellular vesicles, here characterized on a Western blot analysis by the marker TSG101, from ribonucleoprotein complexes that are also known to stabilize RNA in different types of sources that are characterized by the presence of the Argonaute-2 protein. So if we were doing a similar analysis in the size-based preparation or the volume excluding preparation, we observed a clear co-separation of these two types of particles. This, of course, indicates that depending upon the method that you use, you will enrich extracellular vesicles with a different level of specificity, what we also refer to as purity of the sample. This is in a very simple biofluid conditioned medium, but the same is actually true if you do biomarker analysis in more complex biofluids such as blood plasma. So for example, in blood plasma, lipoprotein particles are the very famous, well-known potential contaminants of extracellular vesicles because they partially overlap in size, but also density with extracellular vesicles. So in this particular case study, we developed a combination of size exclusion chromatography with density gradient centrifugation. We also decided to compare the protein content of different preparations of this workflow to observe what is the impact of the choice of method on EV analysis starting from a blood sample. So first, we loaded our blood plasma on top of a size exclusion chromatography column, and this typically results in a separation of extracellular vesicles from high-density lipoprotein particles. Then we collect the fraction that contains the extracellular vesicles, and we load it on top of a density gradient. And this density gradient will separate extracellular vesicles from lower density lipoprotein particles. So for this proteomic analysis, we then decided to compare the sample that elutes from the size exclusion chromatography column to the sample that we find on top of the density gradient, which would mainly consist of lipoprotein particles, and compare it to the sample where we presumably identify enriched extracellular vesicles. And if we look to the protein content, we again observe that it's significantly different between the EV enriched fraction that we observe, identify based upon the density gradient, compared to the top fraction of the density gradient, and compared to the protein profile that we retrieve from the fraction that elutes from the size exclusion chromatography column. And what is very striking here is that actually the sample that elutes from the size exclusion chromatography column resembles more the lipoprotein fraction than it does resemble the extracellular vesicle fraction, meaning that the level of lipoprotein contamination upon size exclusion chromatography is substantially high and thereby partially masks extracellular vesicle enriched proteins. Now, it's not only a consideration in terms of biomarker analysis, but the choice of method can also impact downstream functional analysis of extracellular vesicles. So in this particular study, we more or less implemented a similar strategy, but instead of preparing conditioned medium from breast cancer cells, we now prepared conditioned medium from cancer-associated fibroblasts. We prepared extracellular vesicles with a density-based method, size and two size-based methods, in this particular case, differential ultracentrifugation and size exclusion chromatography. And we assessed the potential of these preparations to stimulate the maturation of dendritic cells. And we actually observed that only the extracellular vesicles that were retrieved with the density-based method, those dependently increase the maturation of dendritic cells in contrast to the size-based methods. Of course, also here we did a further assessment to identify the reasons of these differences between the isolation methods, and we noticed that in contrast to density gradient centrifugation, differential ultracentrifugation and size exclusion chromatography did not efficiently deplete soluble cytokines that were present in these samples and that also have an immune inhibitory role or function. Not only methodological heterogeneity exists at the level of isolation methods, so methods that you use to prepare vesicles from the biofluid of interest, but it already even starts with pre-analyticals. So in this particular case, we were interested in analyzing extracellular vesicles from blood plasma samples, and instead of wondering which preparation method we should use, we were also wondering which blood collection tube should we use, and thus the choice of blood collection tube or the processing time of the blood sample, for example, impact the downstream analysis of extracellular vesicles. And I will not go in too much detail here, but the answer to this question is yes. So depending upon the blood collection tube or the blood processing interval that you implement, you have, for example, a differential activation of platelets, a differential contribution of hemolysis, red blood cell-derived EVs, for example, that all impact the downstream biomarker analysis of extracellular vesicles. So these few case studies as an example to underpin that the choice of methodology is crucial for a downstream extracellular vesicle analysis, not only at the level of preparation method, but also at the level of pre-analyticals that can impact different steps of your workflow. Now, building upon this interest in the impact of methodology on downstream extracellular vesicle analysis, we then decided back in 2014 to do an in-depth analysis of literature to see how methods are actually being reported in the field of extracellular vesicles. And there too, we analyzed about 1,200 publications for 115 experimental parameters. And these experimental parameters questioned the preparation method that was used and also the characterization that was performed on the obtained preparations, being both at the protein level and the particle level. And what we actually observed is that those more than 1,200 publications report more than 1,000 unique protocols to obtain extracellular vesicles from different types of biofluids. And besides that, also almost 20% of these publications did not perform any downstream characterization of the preparations that they obtained. So this really illustrated to us, so this work was first published in 2017 based upon a literature analysis between 2010 and 2015. It illustrated, first of all, that there is a huge methodological heterogeneity, but that there is also a lack in transparency in reporting the results of these studies. So I think we are all very well aware that different initiatives since then have been made with the aim to really steer transparency and reproducibility in the extracellular vesicle research field. The very first effort was made in 2014, where a first initiating set of BESEV guidelines was developed and released to the community. In 2017, we released the EV-Track initiative with the aim to develop or provide a tool that really helps EV researchers towards implementing transparency. And after that, multiple updates on the BESEV guidelines have been established. The Rigor and Standardization Subcommittee of ISEV has been launched, and multiple position papers dedicated to particular technologies or particular sources have been released. And of course, all these efforts are there and really help to increase reproducibility in the field. But I think the overall reason of why all these initiatives were taken in the meantime is because overall, of course, there is a need for a supporting ecosystem, because in the end, the reason why we study extracellular vesicles is because we want to develop them as types of applications that can advance multiple research domains. So based upon the literature study that we did, we decided then to develop the EV-Track knowledge base as a tool that can be really helpful to guide the research field. And first, I want you to explain how we initiated this initiative. So I indicated that we performed a systematic literature analysis. It's not easy to read over 1,200 publications and map different types of experimental parameters. So we decided to set up what we refer to as the EV-Track Consortium. So we had 39 institutions involved in these initiatives, including 95 participants, ranging from early career stage researchers up until principal investigators. We all together screened these manuscripts for all the parameters, and then we were looking for an approach that would enable us to easily understand what the needs of increased transparency are in the EV research field. And therefore, we decided to develop the EV metric. And the EV metric is a measure for the level of transparent reporting in extracellular vesicle research. So I indicated we mainly performed the main analysis in 2015. So we decided to establish this metric and build it upon the BISEV guidelines that were available at that time point, which were the BISEV guidelines from 2014. And together with the EVTREX consortium, we came up with a set of 9 experimental parameters that we thought were really critical to be able to interpret experiments that are reported in literature and to also reproduce them. And these parameters related to the analysis at protein level. So for example, 3 or more EV-enriched proteins should be analyzed, 1 non-EV enriched proteins. Electron microscopy should be performed, revealing both close and wide field electron microscopy images. A density gradient can be used as to validate the observed results, and also some reporting when ultracentrifugation is used, report on all the specifics and experimental parameters that are associated with this preparation. For protein analysis, really report the antibody that you use, the concentration of the antibody that you use, and so on. And also the lysis buffer composition. So all really detailed parameters that we were looking for on how transparently these were being reported in the field based upon the BISEV 2014 guidelines. And what we then actually observed by evaluating this EV metric as a component, as a metric consisting of these 9 components, we observed, for example, that the majority of studies, or at least a significant amount of studies, received an EV metric of 0%, meaning that they did not report on any of these 9 components, although they are reporting results on extracellular vesicles. And actually, none of the studies back then was able to reach an EV metric of 100%. In this spider chart, we can visualize the different 9 components, and it really clarifies where there was a lack in implementation or transparency in reporting these 9 components. Overall, most reported and implemented was the analysis of EV-enriched proteins, but still this was only done for 50% of the manuscripts that we analyzed. Analysis of non-EV-enriched proteins, performance of qualitative and quantitative particle analysis, for example, really were not performed in the majority of, or only in a minority of the publications. A very simple one to report, antibody specifics also largely remain underreported. So this stimulated us to actually use this knowledge to really develop a platform that increases the awareness of important parameters to EV researchers that really matter in the EV research, and in other words, what are the parameters that really can have an impact on your downstream analysis and should be transparently reported so that other researchers can reproduce your experiments. So we developed a knowledge base that consists of 7 enabling features. They are all highlighted here on the circle, and I will divide them into 3 sections. The first section are 3 enabling features that relate to stimulating transparent reporting. This means that we ask researchers to upload their manuscript prior to submission to a journal for publication. In return, the researchers will receive an EV metric, and based upon the EV metric, they can use this as a coaching tool to increase the level of transparency in their material and method section. So how does this work? First of all, you go to the EVTREC website, and there you can create a login. So you develop a My EVTREC account, which is just requesting an email account and a password. And then you get access to a survey where you can upload your unpublished experiments. And so to clarify, if you have your manuscript ready for submission to a journal, You basically have a look at your material and method section, and you complete survey questions based upon the information that you wrote down in the material and method section of your manuscript. Once you have completed the survey, you will receive an EV-Track ID and a preliminary EV metric. And why is this a preliminary EV metric? Because your unpublished experiments will be in a closed database, which is only accessible to you and to people with which you share the entry code. So for example, what we stimulate is that you share this entry with journal editors and reviewers so that they are aware that the level of transparency and reproducibility in terms of experiments on extracellular vesicles is high and can be guaranteed through means of the EV-Track knowledge base. But it's of course closed because only after publication we will curate the data that you have submitted to the knowledge base. Once it is accepted, we will also link the entry to the PubMed ID, and then your manuscript, at least the parameters that you submitted to the EV-Track knowledge base, will become publicly available together with the EV metric. So how does this look like? If you go to the EV-Track knowledge base website, you will find on the right tab the My EV-Track link. You can click on it. You have to log into your account, and then you can get access to different types of surveys. The first one will be the most important one, because here you will be able to upload new data. But let's assume that you already published an experiment and you observe, and it has been submitted to EV-Track and made available on the EV-Track knowledge base. It is possible that you observe that you have some information available, but that you did not write it down in your manuscript. Then there is the possibility to also add additional information to your entry to further increase transparency and reproducibility of your manuscript. But for today, let's focus on the upload of new data. So if you click that button, you will get access to a survey. And with this survey, it starts with very broad questions, but most of the time it will be a pre-publication submission. So prior that you submit or publish your work in a journal. However, you can also do this post-publication. So let's say after the lecture of today, you say, hey, we have this EV research publication. I also want to submit it to EV-Track. It's definitely possible to still also edit post-publication. However, the only disadvantage is that you cannot increase anymore the transparency and reproducibility by changing the material and methods of your published work, but you could still add this information to the EV-Track knowledge base. So then you go over the whole survey. Depending on how your protocol and setup looks like, it will be a maximum of about 300 parameters or questions that you have to address. The 300 is the maximum, so you will not implement each of the methods that we potentially question in the survey, for example. You will not also use all the different types of biofluids for which we have survey question. So typically, you will complete fewer than 300 parameters, but depending on your experimental setup, the number of questions that you will have to address during the survey, of course, will depend. Once you complete the survey, you will receive a report containing the EV metric, consisting of 9 components that are indicated in green if you correctly reported them in your manuscript, and in red if you did not report it. And what we actually observed is that in more than 80% of the experiments, the EV metric is increased by increasing reporting of parameters rather than having to perform additional experiments. So one aim of EV-Track stimulate transparent reporting. Second aim is, of course, also centralize the knowledge on EV biology. For example, we also request information if you did a particle analysis We can, for example, you can add information on the average size or the mean or the median size that you measured, particle concentration, but also density. So it really allows us to capture information on EV biology. So also which type of proteins that you analyze, for example, and so on. But we also get information on which methods are, for example, used for which types of biofluids, and all this information can be queried by the end user. So how do the search functions look like? So for now, this is still a little bit limited, but basically any of the parameters that you would be interested in can be queried through the knowledge base, through the search tab, and you can also download the data and perform downstream analysis, making use of these data that are available on the EVTrack knowledge base. So in the end, it is important then, why should you use EVTrack, or why is it important to use EVTrack? First of all, in terms of transparent reporting, in my lab, for example, we use it as an education and a training tool. So any master, PhD, or postdoc that is entering the lab has to explore the tool and implement it. And why? And anybody after the training will say, well, it really learned me which different parameters are important to consider when you study extracellular vesicles. So as an educational tool, and really to understand for which biofluids, which parameters should be reported, or for which method, which parameters should I consider when I perform this method, is really valuable information when you're setting up protocols, but also writing down your experiments into a manuscript. Of course, by doing this pre-publication, if you receive an EV metric, you also have the possibility to make your material and method section as complete as possible, so that you increase the transparency and thereby also the potential reproducibility of your work. Because if you have worked for a couple of years towards a manuscript, if you publish it, of course, you want other people to be able to reproduce and confirm the results that you obtain, because this is what we need if we, for example, want to validate biomarkers or therapeutic applications. But also by submitting to the EVTrack knowledge base, you can also increase the visibility of your work, because upon publication, the entry will become publicly available through the EVTrack platform, which is highlighted also on the platform, for example, and also by means of our Twitter account and social media. As most of you probably know, it's also one of the BESEV recommendations, and EVTrack is also endorsed by the International Society for Extracellular Vesicles. And by doing so, you really also contribute to keeping EVTrack a living knowledge base, which is of course crucial if we wanted to accurately capture our knowledge on extracellular vesicles in terms of biology and methodology. Why should we use it, or how can you use it to centralize knowledge? Well, as I indicated, you can develop multiple queries. But what we, for example, typically do is when we write review or research manuscripts, or we have a particular research question, we search the EVTrack knowledge base to gather an idea about the current state of the art. So I highlighted the study where we compared different blood collection tubes. Well, one of the first things that we did was access the EVTrack knowledge base and get an idea on which type of blood collection tubes are currently being reported. And this really allowed us to set up this study. Which blood collection tubes should we compare and measure, and which are the most commonly used ones to study extracellular vesicles in blood? Then you can also observe that 50% of studies do not even report the blood collection tube that they use. So these are very interesting analyses that can be easily made starting from a query of the EVTrack knowledge base. Similar, we did it for urinary EV biomarker discovery, where we screened all urinary EV papers that were interested in the development of biomarkers for their EV metric to assess the current state of the art in the urinary EV biomarker field. Besides that, you can also use it for systematic reviews. So here's an example of another research group by Vet Holder, where they implemented the EV-Track knowledge base to map the current state of the art in studies that are analyzing pathological pregnancies, for example. So for all the different studies, they reported also the different experimental parameters by submitting these studies to the EV-Track knowledge base. So it's a very convenient tool to develop these queries and to really assess for your particular study case what is the current state of the art. Also, when you start to explore a new biofluid and you do not really have an idea which method to use to prepare or to characterize the vesicles, you can implement the EV-Track knowledge base to start the query on commonly performed methodologies on this particular biofluid. Now, as I mentioned, it is important to keep the EV-Track knowledge base alive. And of course, since the publication or its release in 2017, we have made multiple improvements to the knowledge base as well. So I indicated in the first study, we analyzed 115 parameters related to the sample type, the preparation method, the characterization method. To date, we are actually over 300 parameters, questioning for each of the biofluids which pre-analyticals are important, and more extended parameters, additional methods that have been added in terms of EV preparation and characterization. But this, of course, the increase in this number of parameters results through the tremendous efforts that have been made by the different ISEV task forces that identified for different types of technologies or different types of biofluids, what are the critical experimental parameters. So for example, they have been reported for CSF, for cell culture, for urine as well, for blood, and then for particular methods such as the MyFlowSite EV. And each of the experimental parameters that are proposed by these ISEV task forces to be really fundamental for that particular source or characterization method have all been integrated into the EV-Track knowledge base so that we really capture the relevant experimental parameters for these different cases. Initially, we analyzed 1,226 publications. To date, we already have more than 3,000 publications in the knowledge base that report more than 9,000 different experiments. So there is a huge increase in the amount of information that has become available in the EV-Track knowledge base. And what is very nice to observe is the evolution in EV metric. So until 2015, we analyzed these papers or manuscripts were analyzed for the development of the EV-Track knowledge base. Remember, in 2014, we had the first release of MySEV guidelines. In 2018, the first community-supported MySEV guidelines. And what we actually observe is since the release of these MySEV 2018 guidelines, there is a substantial increase in the EV metric, indicating that the development of these guidelines really stimulates the improvement of transparency and reproducibility. What we also observe is that there is an increase in the number of manuscripts that achieve an EV metric of 50% and higher. So it means that by providing these tools and these guidelines, that the level of transparency is gradually increasing in the EV research field. If we then take a look to the overall EV metric, we do observe that there is also a very well increased improvement in reporting, for example, lysate preparation specifics, antibody specifics, and ultracentrifugation specifics. It's almost 90% for most of these. But we also observe that for some of these aspects, there is still a delay in the increased level of transparency. So these are aspects that we can then still focus on to see how guidelines should develop and how we can further stimulate transparency and reproducibility for these different components. And of course, the user committee of the EVTrack knowledge base has also increased. So we started with 39 institutions. Today we have more than 900 institutions making use of the EVTrack knowledge base with more than 1,400 accounts registered. In terms of coaching tool, we have also tried to really implement and merge EVTrack into different other types of research databases, such as Vesiclepedia, for example. So we developed an EVTrack summary add-on that allows to link or to establish a link between the EVTrack knowledge base entry and the entry of the same study in, for example, the Vesiclepedia knowledge base, which is of course crucial if you want to do an in-depth analysis of publicly available data in terms of RNA or protein composition that are reported in these databases. It is of course also very, very valuable to know which methodology was used, how transparently was this methodology reported, and so on. So this has been made available also by the development of this summary add-on. And then a final advancement that we are making is to reassess the composition of the EV metric. So initially it consisted of 9 components, where actually 5 out of 9 components refer to the interpretation of data and 4 to the reproducibility of data. In terms of reproducibility, we now slightly propose an adaptation in where we do not only request on the specific reporting of a couple of parameters related to protein analysis or the use of ultracentrifugation, but we adapt the metric by just assessing the report of each of the different parameters that you should report if you implement a particular method. So for example, in terms of pre-analytical variables, if you work with blood plasma and you were to report 10 parameters, you were to report 4 parameters and you reported all of them, you would gain an EV metric completion of 100% for that part. Here only 50% is reported, for example. Similar for the separation protocol and also the molecular characterization and the particle characterization. By doing this, we aim to further stimulate the use of the EV metric as a transparency tool to really stimulate researchers to report any of the experimental parameters that should be reported for this particular set of methods for preparation, characterization, and also in terms of the source. So overall, EVTrack knowledge base has had a significant development during the past couple of years, where we are working towards the implementation of the new EV metric format. We established it as a coaching tool. We stimulate the use of the knowledge base so that we can really capture the trends that occur in terms of EV research and biology. In close communication and interaction with the ISEV task forces, we have also updated the survey with relevant parameters for different sources and methods. And most importantly, also the survey that is used to upload experiments to the EVTrack knowledge base has been updated and will also soon be released. So the manuscript is currently in preparation, and the release of the second version of EVTrack is also under preparation, and we hope to release it by the end of this year at the latest. The final part that remains is a community. And it's of course you that really decides on the future of the EVTrack knowledge base. To keep it living, we need your contribution and we need your submissions to the EVTrack knowledge base. But I also mentioned that the EVTrack knowledge base is now endorsed by ISEV, and we had the opportunity to establish an ISEV rigor standardization task force, which really has to define the future of the EVTrack knowledge base and also is responsible for the maintenance of the EVTrack knowledge base for the future. So how does this work? Well, basically, we have established the EVTrack task force, which includes the participation of early stage researchers together with principal investigators that are tasked with, first of all, maintaining and implementing future improvements to the knowledge base, but also maintaining close interactions with each of the different task forces to ensure that the knowledge base and the survey are up to date with the current needs so that it very accurately can capture the current state of the art in EV research. So as a take-home message, we have to consider that the analysis of extracellular vesicles in different types of biofluids is very challenging, and that we cannot circumvent methodological heterogeneity because different sources, different study aims will require different methodological setups, and this heterogeneity is also fundamental to enable discoveries. But with EVTrack, we really aim to unite and support EV researchers to provide guidance towards transparency and reproducibility. And of course, this transparency and reproducibility, by means of EVTrack, we aim to make this a very small step for an EV researchers, but we hope that it results in a giant leap forward for the EV research field, because overall transparency and reproducibility are key to really develop a reproducible ecosystem that can enable applications of extracellular vesicles in different research domains. So I hope that I convinced you that you should really visit the webpage of EVTrack, register an account, and start to submit your work to the EVTrack knowledge base so that we can help you in transparently report the work, that we can maintain the knowledge base and also make it up to date for future query functions and support you like that as well. With that, I want to thank the EVTrack consortium, of course, the EVTrack administrators that have been very active in maintaining the EVTrack knowledge base and also interacting with researchers to address their different questions during their use of the EVTrack knowledge base, which are Quentin, Sarah, Sander, and Jan. I also want to thank the different EVTrack consortium members and of course all the users that we have had so far. We look forward to welcome many new members and users in the future, but I also want to thank ISEV particularly to allow us to establish a task force that really allows us to guarantee the future existence and development of the EVTrack knowledge base. With that, I want to finally thank my group, my lab members, my funders, and thank you for your attention. And I would be happy to take any of your questions.
Thank you so much. That was wonderful. I'm going to hand it over to Yanis to handle the questions.
Thank you very much. Yes, again, thank you again, Professor Hendrix, for the amazing talk. It was very, very useful. So there are already some questions in the chat, so I'll start with those. So the first one is from Yoon Lim. She says, great talk. I have a question about urinary EV isolation. Is creatinine the best molecule to normalize EV concentration isolated from urine?
I think there is not always one best option, as we know for many particular aspects of EV research. But creatinine, I think if you would look to the dedicated guidelines that were developed by the urine task force from BESEV, I think creatinine is indeed one of the potential parameters that you can use to normalize your data, definitely, for sure.
Thank you. And just to follow up on that question from another user who asks, what would you suggest the best protein to normalize large EVs or microvesicles immunoblotting isolated from human plasma?
Yeah, that's a similar. I think there is not one best option. It of course depends on. So this was for blood plasma. Sorry, I missed the last.
She just says human plasma, so I'm assuming yes, it must be blood plasma. But
Okay.
if I'm wrong, they'll mention in the chat.
So in terms of normalization, I will basically just share how we do it. So once we isolate our vesicle from the sample, we do a particle concentration measurement and a protein concentration measurement. And then based on what you really want to analyze, we load same amount of particle or same amount of protein on our Western blot membranes. And then we do an analysis of EV-associated proteins and non-EV-associated proteins. So in terms of which protein would now be the best, I really would not like to make a recommendation because this really depends on what you're looking for. I think it's just important to normalize the data that you want to compare, but I think what could work as well is that you load same amount of protein concentration or same amount of particle concentration. I don't think there is one key protein that you can load just to show that you have a particular loading control. I know some use CapDH, for example, but this is something that we classically not do. We work with particle or protein concentration.
And just to follow on that, the user's asking, when you use particle concentration, we assume it's by NTA or with nanosite or ZetaView or something like that?
Yes, we use nanoparticle tracking analysis, which has its limitations because we are also looking to smaller vesicles below the 17 nanometer detection range. So for every method that you use, you should be well aware about the limitations of the methodology and really try to decide what would now be the best way to normalize for the data. But for an average EV population, NTA can work. If you're particularly looking for small EVs, NTA is not a technology that I would recommend.
Thank you. So another question from Sandra. She asks, what is your recommended method of EV isolation? And then also, what's your opinion on differential centrifugation and filtration to isolate blood EVs?
Again, there is not one size fits all solution. I do not have a recommended method because it's very difficult, but I do have clearly a preferred method that we use in our research group, which is always density-based. And why density-based? Because we mainly focus on biomarker discovery. And I'm really convinced if you want to discover biomarkers that you have to purify your vesicles to the extent that you do not have abundant lipoprotein contamination in blood, for example, or Tam-Horsfall protein complex contamination in urine samples. And there, we have different publications showing that you really need a BEV preparation of high purity to be able to enable this biomarker discovery. And this is always by exploiting two different types of biophysical characteristics. So I think instead of recommending which type of method it is best, I would rather recommend if you have a complex biofluids like blood or urine, likely with one method you will not achieve a good level of purity, but make use of a combination of methods that exploit different characteristics of extracellular vesicles. So for example, combine a size exclusion chromatography with a density gradient, or combine a size exclusion chromatography with an immune capture, but try to exploit different features of extracellular vesicles. This will stimulate purified enrichment, but it also comes with the consequence that you will have a lower recovery efficiency, so you may lose some of the vesicles. But I think depending upon the application, and if it's towards biomarker discovery, purity is more important than recovery efficiency. And if you go for purity, try to combine different methods. In terms of differential ultracentrifugation and filtration, I see filtration as a concentration step in combination with differential ultracentrifugation, and differential ultracentrifugation is enrichment step. So we never use filtration as a sole isolation method, but we use it, for example, to concentrate or conditioned medium, or to filter out dead cells or free floating cells that could contaminate the preparation.
Okay, I will ask a question about EV track, because I think a few of the questions we have are mainly to do with EV isolation. And so my question is, you mentioned during your talk that with EV track, it helps with with reviewers from journals to help give a nice review about papers that are undergoing through publication. How easy is that for journal reviewers to go through? I mean, outside of traditional EV journals such as JEV, JX Bio, have you had problems with certain journals that they find it a bit more difficult, it's a bit more hurdles, or are they more accepting that we have generated this database to help us kind of have a bit more regiment about what we're doing?
Yeah, my general idea is that journals really like it. But it's really up to the researcher to guide the editors and the reviewers to the link. So there is a reviewer-editor link tab on the EVTrack knowledge base. And if you provide them with the access code, they can then access the entry. And the overall feedback that we receive from journals is very positive, even beyond the field-specific journals. So also the Nature Publishing group is very active in using this knowledge base, for example. The most difficult thing is it's more difficult to ask journals to request researchers to implement the use of the EVTrack knowledge base, because then you have to go to the publishers and to negotiate the implementation of EVTrack at that level is very hard for the moment. But most of the times, what I commonly see is that many reviewers and editors actually ask authors to please upload your experimental parameters to the EVTrack knowledge base. And this is something that we did not notice in the first years when we released the EVTrack knowledge base, but now it's common practice, and not only for reviewers from the Journal of Extracellular Vesicles, but also from other journals. So we see more implementation also for different journals. Most of the time through the reviewers and the editors that make the request to do this.
Yeah, I have a question myself, if I may. You mentioned that early career researchers can be involved in the EVTrack consortium. And I was wondering if you could elaborate on the process of getting involved, how this works, who should we approach, how to get involved and help with the growth of this very useful platform?
Yeah, so well, there are two aspects. First of all, unfortunately, for now, we completed the composition of the task force. So I think we launched a survey in last summer, and we are now actually training the task force members in using and maintaining the EVTrack knowledge base. So for now, to really participate as a member of the EVTrack task force, unfortunately, it's a little bit too late at this stage, but we do foresee a regular turnover because we do feel that it's the early stage researchers that are really the main users of the platform. So we definitely would value, if you are using the platform and you have any recommendation or aspects that you encounter that think can require further improvement, yes, please reach out to the EVTrack knowledge base, and we will also consider this as an interest from your side to be considered as a future task force member. So I don't know at this stage yet how soon we will do a turnover. It's also possible that we will need an additional amount of members to keep up with all the work. For now, we have 50 members involved. I think half of them would be more or less early stage researcher. So it's one of the only task forces of ISEF that has such a high participation rate of early stage researchers. So please, if you have interest, when you're using the EVTrack platform and you have suggestions, reach out to the task force chair, and that would be me, or any of the other task force members that are involved. They will be listed soon, I think, on the ISEF webpage of the Rigor and Standardization Subcommittee.
No, thanks. And just to follow up on that and the previous question, with EVTrack trying to be implemented more in the community, but also with journals and reviewers, a member of the audience asking, is EVTrack going to be mandatory in the future for publishing to high-profile journals such as JEV or other things like that?
Yeah. Mandatory is a difficult word. I think if you make something mandatory, you start to dislike doing it. So I don't like really to make it mandatory. I would like people to understand all the benefits of the EVTrack knowledge base, so that it's a pleasure for you to make use of the knowledge base. So if they would make it mandatory, we had this discussion within the Journal of Extracellular Vesicles already. And as I mentioned, we do many times observe that reviewers ask to authors to submit their parameters, and authors do it. So I think it will never become mandatory before you submit a manuscript to a journal, but it can, of course, be requested by reviewers or editors. I think like mandatory for me always has a negative connotation. So I would try to avoid to make it mandatory, but also with the ISEF task force, we are searching for different ways to really reach out to the community and to really stimulate the implementation of the EVTrack knowledge base, but from a positive perception rather than from a mandatory perception. Yeah. But you
So
will
not mandatory, but strongly recommended.
Yeah.
Okay, so I have one more question. This is from the president of the SNEV community, Sarah. She asks, if you could only choose 2 parameters for people to report on EVTrack, what would, in your opinion, what would be the most important?
I would recommend to report all the parameters that have been suggested by the different ISEF task force committees. But then I would think if we really had to select 2 parameters, I would, first of all, so they are divided over 2. You have the ones related to interpretation of data, and the other one really like, for this set of experiments, you have to report this set of parameters. Did you report them all or not? Then for me, there are 2 important aspects. First of all, did you report all the parameters? That is crucial, because whatever finding, if you want other researchers to validate it, you need this transparency. And then secondly, validate your results with another method or using proper controls. I think that would be for me the 2 most important aspects to consider.
Okay. I don't think we have any more questions, and I think it's 2 o'clock, so maybe we should call it time. Once again, thank you very much, Professor Hendrix. Thank you very much for the talk and for your time. Thank you, everyone else in the audience, for listening and for your questions. And yeah, thank you very much.
Thank you. Bye.